So we are looking now at the photosynthetic equation. Carbon dioxide is going to be used in this process, as is water. We don't just take carbon dioxide and water and smash them together. That's sort of what it looks like by this equation, because you can account for all these things, right? We have carbon, we have oxygen, we have hydrogen. All of those together we can make sugar out of and make oxygen. But the carbon is going to be used here for this sugar. Water, we are just going to steal some electrons from. So these hydrogen and oxygen molecules won't necessarily be used in this particular sugar molecule that gets made. They'll have an important role. The oxygen will be released, so this oxygen does come from this water. It's a waste product in photosynthesis. Plants can still use it to do cellular respiration, just like we do, but most of that oxygen, about 90% of it, is actually just going to be released into the environment for other organisms to then use for cellular respiration. The water is going to donate its electrons, and then these hydrogen atoms are going to be used to form like a battery within the chloroplast, which we'll get to. And that battery is then going to provide the energy to build these complex, larger sugar molecules. One of these sort of ideas in chemistry, not ideas, concepts that you should understand, is that building molecules costs energy, because as you build that molecule, you're sort of forcing things together. Some things want to come together more than others, like if they have positive and negative attractors on them. That whole process is going to result in this sharing of electrons, and that sharing of electrons in this sort of complex structure that has a lot of order to it has energy stored in it, and that's chemical energy. So the energy that we put into building it gets stored in there as chemical energy. But we can't just build these molecules without some sort of energy input. So that energy input is going to be from light and from destroying these water molecules. So what is light? Light travels as both a wave and a particle. It's this wave-particle duality that it has. So we can think of it both in this form, this wave form, and the frequency it has. We can also think of it as these individual sort of packets or particles of energy, and we call those photons. It sounds a lot like proton, but it's different. So that's a different particle that we're talking about. So a photon, like photosynthesis, is a distinct sort of energy packet. And when we talk about light, often we're going to use both of these ideas, this idea of a frequency as well as this idea of a distinct little packet. When we look at the frequency, light traveling as a wave, that is going to determine sort of how much energy that bit of light has. So if we break light up into different components, we can get these different frequencies out of it. And as light travels from the sun into our atmosphere, it's going to hit different particles and get scattered. Some of those wavelengths are going to get reflected back. Some of them are going to make it through. Lucky for us, our atmosphere blocks and reflects back some of these more intensive energy light waves that would maybe destroy our DNA molecules and render us mutinous blobs of fleshy goo. But these ones that we see, and some a little bit beyond our sight in both directions, do make it through. So this tiny fraction right here represents the visible light that we see. And we distinguish that as different colors. That's how our eyes interpret it. And each of those different colors represents a different frequency of wavelength. The higher frequency wavelengths have more energy, and those are at the purple and blue side of the spectrum. The lower energy wavelengths are the reds and oranges, and that's at that side of the spectrum. So we talk about light as colors, we talk about it as energy, we talk about it as wavelengths and photons. All of those mean the same thing, but are different ways of talking about it. Why do we need to understand this? Because photosynthesis, we're going to be talking about pigments. Pigments are these molecules that reflect particular wavelengths of light and appear a certain color. Now, a lot of molecules will just sort of appear transparent, like a lot of proteins. When you put them into a glob that's large enough to see, it just doesn't really reflect any light at all or absorb any light. It just is transparent. It lets the light go right through. So like albumin, your egg whites, don't have that pigmented property. But other compounds, like the anthocyanins you're going to look at this week, or like the chlorophylls and carotenoids that you're going to look at in your thin layer chromatography, these are all molecules that have a shape that absorb some wavelengths of light and reflect others. So it's important to understand that the color that we see is the light that's reflected back at us. So those are wavelengths of light that those particular molecules are not using in any way, but they're reflecting back. So chlorophylls are molecules with this overall structure. It's got this kind of like squarish shape with the little fatty acid tail, and there's a lot of nitrogen and a little bit of magnesium in here. So plants need a lot of nitrogen to make chlorophyll. These molecules reflect green light, and there are, I think, 5 different chlorophylls. We have looked at A, B, and C, but there's also D, and then there's this bacterial chlorophyll, which is pretty similar. So they are reflecting the green light. That means that that energy is not being used by those molecules. We've talked about this a little bit as we talked about different pigments in evolutionary groups. So we can look at this absorption spectrum and see that these peaks represent the light waves that are being used. So that's energy that that plant can access from that light. So we can get a lot of these violets and blues from chlorophyll B, as well as a little bit of the orange. We can get a lot of blues from carotenoids. And for chlorophyll A, we get more of the oranges and violets. So that's kind of the important part for plants. For us, we sort of think about this in reverse as what we see from those. So these troughs here represent the kind of spectrum of light that gets reflected back at us. So chlorophyll A reflects back more of this blue-green, while chlorophyll B reflects more of this yellow-green. And that's what they look like. So you can distinguish them on your thin layer chromatography strip by their color, but you can also distinguish them by their polarity. Here's an example of a very well-developed thin layer chromatography strip. Yours may or may not look like this. It will most likely be less orderly and have a kind of pointed, weird, hard to figure out structure to it. This one's great. It's got these nice lines defined. Don't expect yours to look like this, but if it does, excellent. So here you can see the chlorophyll A is a little bit more blue-green. This chlorophyll B is a little bit more yellow, grassy green. But they're in different positions. Why does that happen? When you do thin layer chromatography, you're extracting the pigments out of the leaves that will come out in that extraction solvent. So another important chemistry concept is that like dissolves like. Non-polar compounds that don't have excess charges on them will dissolve other non-polar compounds. Polar compounds will dissolve polar and ionic compounds, so things that have a lot of excess charges. The two are generally not going to mix with each other. So if you put water and oil together, water is polar, oil is nonpolar. They don't mix unless you have something that's there that's got a polar part and a nonpolar part that can bind them together. But even then, it's like a tenuous bond, right? So to extract these pigments, we are using a nonpolar solvent. So we are only going to really get nonpolar compounds out of it. And there's a spectrum. Things aren't just polar or nonpolar. They're more or less polar, basically, or more or less nonpolar. So any compounds in your leaves that are polar compounds, strongly polar, are not going to be extracted in your extraction solvent. If they do make it into your little pigment dot, they're still going to get left at the bottom and stay at the bottom. So anthocyanins are an example of a pigment that is polar and will not show up in your TLC extraction. It won't travel with the solvent. So you've put your little pigment dot there, and now you're letting solvent carry the pigments up as the solvent travels up the paper. And it'll carry those pigments up with it. The stronger those pigments are bonded to that solvent, the better dissolved in there they are, the higher they'll go. So this allows us to separate pigments based on their polarity. Compounds that are more polar will stay closer to the bottom of your strip. They won't travel with the solvent because they are not really that attracted to the solvent. So they're not going to travel up with it. The more non-polar your pigments are, the higher they'll travel. Actually, often beta-carotene, which is extremely non-polar, will travel all the way up to the very edge of your solvent front. So sometimes you'll have a faint orange line up there, and that's your beta-carotene. When you look at these, you are hopefully going to be able to do some identification based on color. Hopefully your chlorophylls will separate apart from each other, and you can kind of see that chlorophyll A, which is also slightly more non-polar, less polar, will be more blue-green. It'll also be higher up. Chlorophyll B should be a little bit lower and more grassy in color. There are carotenoids that are orange that will travel all the way to the top, like beta-carotene. There's also phaeophytin. There's compounds called xanthophylls that are yellow that are more polar, less non-polar, and those will stay closer to the bottom. Lutein, I'm pretty sure is a carotenoid, but now I feel like I should probably look it up. But that one generally stays just below the chlorophylls. So as you separate these out based on polarity, you're then going to measure how far they've traveled, calculate something called an RF value, and then use that to try to identify the pigments. But you're probably going to mostly be using position and color to get an idea, and then use the RF value to try to find a more specific example. Okay, so I'm going to get to this next part in the next video.